My name is Zidong Zhang. I'm from Princeton University. Today I will share our work on developing a computational framework for studying gene regulation mechanisms from single-cell multi-omics assays. From our prior knowledge, we already know the existence of multiple mechanisms involved in the regulation of gene expression, such as the regulation from cis-regulatory elements and the transcription factors. Our goal is to understand the mechanisms of regulation for each gene in a data-driven manner by analyzing large datasets of different types of genome-wide assays. This data-driven approach has become more and more powerful recently because of the development of single-cell sequencing assays. However, previously developed computational methods usually uses only one type of single-cell data, such as single-cell RNA-seq or single-cell ATAC-seq, and it's not easy to combine the inferences from multiple types of data. Recent development of single-cell multi-omics assay that run RNA-seq and ATAC-seq from the same single cell provided new opportunities for studying gene regulation more comprehensively. So we developed a new computational framework specifically for studying gene regulation from single-cell multi-omics assays. We call it CREMA, which stands for Control of Regulation from Multi-omics Assays. The goal of CREMA is to combine the same cell measurements of RNA and ATAC to extract mechanisms of gene regulation with high quality. The input data to the framework is single-cell multi-omics data, where RNA-seq and ATAC-seq are simultaneously measured in the same cell. We first select candidate regulatory regions, including both the proximal region and co-accessible distal regions, and we use motif analysis to find potential binding sites of TFs in these regions. Then in the model building step, we use a linear model to select TFs that not only co-express with the target gene, but also has accessible binding sites in the regulatory regions. The output of the framework includes both the regulatory networks as well as the regulatory regions that these TFs bind to. We first evaluated CREMA's identification of distal regulatory regions by checking the enrichment of EQTLs, because EQTLs indicate locations on chromatin that might affect gene expression. We checked not only statistically associated EQTLs, but also fine-mapped EQTLs from 3 fine-mapping methods. In all cases, CREMA shows enrichment of EQTLs compared to a baseline method that only uses co-accessibility analysis from the ATAC modality. We also evaluated CREMA's reconstruction of regulatory network by measuring how it recapitulates gene expression patterns in independent test datasets. We compared CREMA to CINIC, a state-of-the-art method that infer regulatory networks from single-cell RNA-seq data. We showed that by combining the simultaneous measurements of ATAC-seq, CREMA has better performance in reconstructing regulatory networks than methods that uses only the RNA-seq. We also want to directly validate some of CREMA's findings, and here is one example. We pulled out CREMA's identification of target genes regulated by the transcription factor GATA2 and the regulatory regions that GATA2 binds to. We then compared mice with GATA2 knockout to wild-type mice and used the differentially expressed genes and differentially accessible peaks to check CREMA's predictions for GATA2. We found that CREMA successfully identified target genes and the regulatory sites of the transcription factor GATA2. We then applied CREMA to understanding the regulation of gene expression patterns, and here is one example. We look at the pituitary tissue and focused on the transcription factor FOXP2, which is highly expressed in multiple cell types of the pituitary tissue. When we look at the expression patterns of the predicted target genes of FOXP2, we found that FOXP2 activated different genes in different cell types, as illustrated by this schematic figure. To understand how this is achieved, considering that each gene might be regulated by other TFs besides FOXP2, we constructed the full regulatory network for these genes, and it's shown in this heat map. We found that although these genes share a common TF FOXP2, genes that highly expressed in one cell type are co-regulated by different set of TFs compared to genes highly expressed in another cell type. The full regulatory map has cluster-like structures where a set of genes are co-regulated by a set of TFs. These clusters are enriched for biological pathways and may represent regulatory circuits that are interesting for further analysis. In summary, we developed a general computational framework, CREMA, for inferring gene expression regulation from single-cell multi-omics datasets. CREMA's identification of regulatory TFs and domains were evaluated by experimental data. We also used CREMA to understand the regulatory map of genes with cell type specific gene expression patterns in the pituitary tissue. With that, I'd like to thank my advisor Olga Transkaya and my collaborators. Thanks for attending, and I welcome any questions.